Everyone, this is Priyanka and today I will going to explain you Griffith experiment. So let's start our topic. There were number of experiments done to explain DNA as a genetic material. I think you all were aware of what is DNA. DNA is a genetic material used to store information and transfer information from one generation to another generation. For all living organism, genetic material is DNA, except few viruses. In 1928, Griffith performed transformation experiment he used two different strain of bacterium Diplococcus pneumoniae The two different strains are virulent strain and the second one is avirulent strain. Virulent strain causes pneumonia and avirulent strain do not causes pneumonia. What is the difference in virulence? The difference in virulence is related to polysaccharide capsule of the bacterium. This virulent strain have this capsule and this avirulent strain do not have this capsule. Okay. Now, the non-encapsulated bacteria are readily to engulf and destroy by phagocytic cell, and virulent bacteria which have this polysaccharide are not easily engulfed, hence they are able to multiply and causes pneumonia. Okay, this virulent bacteria have this capsule. That is why it is not easily engulfed by phagocytic cells. Hence, they are able to multiply and cause pneumonia. When then Griffith grow these on an agar culture plate, he noticed encapsulated bacteria he noticed encapsulated bacteria from smooth colonies whereas non-encapsulated bacteria produce rough colony. Encapsulated bacteria produce smooth colonies and non-encapsulated bacteria produce rough colony. So each strain of Diptococcus pneumoniae have different serotype. He used two different strains, that are 2R and 3S. This R1 is rough in morphology and it is smooth in morphology. Capsule is absent in 2R and in 3S capsule is present. This 2R is avirulent and this 3S is virulent. Virulent which causes pneumonia, avirulent which do not causes pneumonia. So what he did, he injected different strains of bacteria into mice First, he took a live cell of avirulent cell of 2R. This is the live 2R. Live 2R. He injected into the mouse and what he get? Mouse survive. Okay. When he used live cell of avirulent 2R, he injected into the mouse and the mouse did not suffer from pneumonia and the mouse will survive. Then next he took live 3S. He took live 3S. When he injected this strain into mouse, the mice will die. So when he used first time live 2R strain and injected into mouse, mouse will survive. The second time when he used live 3S strain, this is a virulent strain, means it causes pneumonia. He injected into the mouse will suffer for pneumonia and ultimately mouse will die. Then he used heat killed 3s. This one is heat killed 3s. This is heat killing. When he used heat kill 3s and injected into mouse, the mouse will survive. In this, when he used heat kill 3s and when induced into the mouse did not suffer from any pneumonia. Mouse will survive in this case. The next, what he did, he used both live 2R, okay, and heat kill 3s. This one is heat killed 3s. Okay, when this mixture of live 2R and heat kill 3s, he injected into the mouse, the mouse will dead. ठीक है? When he used live 2R and heat kill 3s, mixture of these two, when he injected into the mouse will cause pneumonia and the mouse was dead. What he noticed, he said streptococcus cell isolated from dead mice were of 3s type. So what he concluded that all the cells of heat killed 3s culture were dead. It was postulated that some cell of 2R changed into 3s type due to influence of death 3s, and he said this phenomena as transformation. This experiment is also known as transforming principle. What he said, this is the conclusion of his experiment. The component of 3s cell which induced the conversion of 2R cell into 3s cell, and he named as transforming principle. But this experiment was not accepted because he were unable to set what is that material which transfer 3s2 cell which induce the conversion of 2r cell into 3s. This theory was rejected because he did not give any hint to identify the transforming component. Thank you for watching my video.